Okay, welcome everyone. We're just gonna give it another minute. It's just about 2:30 p.m. Eastern, and then we'll begin this afternoon's webinar. Thank you for joining us today. Okay, I'm gonna share my screen and we can get started. Go. All right, thank you. Thank you everyone for joining us today. This is the Spring 2023 COVID Information Commons Research webinar. My name is Lauren Close and I'm the Operations and Communications Manager for the Northeast Big Data Innovation Hub here at Columbia University. I'd like to introduce you to all of the COVID Information Commons, or the KIC. We are a COVID-19 research collaboration platform brought to you by the Big Data Innovation Hubs and funded by the National Science Foundation's Convergence Accelerator. The KIC brings together scholars from all over the country and around the world to share their research findings in the form of lightning talks. And I'm going to drop in the chat just now our website where you can learn a bit more about the KIC. Today, each of our 5 speakers will engage the community directly, answering your questions about their work during a Q&A session at the end of the presentations. I should also note that we at the KIC are committed to providing a supportive and welcoming environment to everyone who works, studies, and interacts with the NEBD Hub community at these public events. So we encourage you to review the code of conduct for our online programming. This includes information about reporting and accessibility features. You can learn more at the second link, which I've just dropped into the chat. But before I hand you over to today's fantastic group of speakers, I'd like to first introduce the NEBD Hub's Executive Director, Florence Hudson, and Florence can give us some background information on the KIC.
Thank you very much, Lauren. And I want to thank Lauren for organizing all of these KIC events. She does a wonderful job as our Operations Communications Manager. I want to thank the rest of the team, which you'll hear about too. So a little bit about the COVID Information Commons. So the history of the KIC, as we call it, is that NSF contacted us in March of 2020 when everything started closing down and people started funding these rapid awards, NSF and I think NIH maybe at the same time, to actually have rapid work done in the research area related to COVID-19. And so they asked us to create a portal for people to easily be able to find NSF-funded COVID research. So at the time, we went into the NSF website and found exactly 32 awards. Since then, the KIC has grown to about 9,500 awards between NSF and NIH related to COVID research and other topics in that area. So we started this in 2020, and at our first meeting, we convinced a couple of PIs to present. So it just wasn't staff people or us presenting. And 2 of the PIs presented and 40 more asked to present. So we started this COVID Information Commons community and told them, until everybody is presented who wants to present, we'll keep doing these. And so we keep doing them because as we go forward, as you know, there's a lot of longitudinal impact of the COVID pandemic and many things we can learn for the future. So we were given an extension award, a KIC extension, we call it, in 2021. And that allowed us to add the NIH awards because we started with NSF. But as we said, this is a disease. We really should be working with NIH too. So we got an extension award. And since then, we've grown the corpus and have, a burst out to the cloud. So we can go to even more awards. Now we're at almost 10,000. So we're continuing down this path. And what it allows us to do is have this COVID PI and awards database, and you can get to it directly from the KIC website, covidinfocommons.net, as you can see on the bottom of the screen. And when you go into the PI and awards database, you can put in keywords, you can choose institution, you can choose NIH centers or institutes, you can choose NSF directorates, state, there are different ways to look at it. And it's a corpus of NSF and NIH research, so we can work in a multidisciplinary fashion. And we update this corpus every month via the NSF and NIH APIs. And we invite students to participate with us too. And we're delighted when they they do our student paper challenges and things like that, which we'll talk more about. So this is one of the facets of the search, which is the PI and awards database. What we also have, and I think Lauren's going to show, is our Lingo4G Explorer tool. So we have, when you look at the main page for the KIC website, you'll see that there's a place for the PI and awards database, and there's a little blue button, I think it is, that has the Machine Learning Maps Research Explorer. And if you go in there, then you'll see something like this. And it takes all the awards and it does some machine learning based clustering into different topical areas. So you or other people can find multiple researchers by topic. You can look at them by PI name. You can look at it by state. You could look at it by institution. It could be you see someone's name and say, oh my gosh, I was a postdoc with them. Where are they now? And you can click and change the view and then figure out where they are, or say, there's somebody else in California. Oh my gosh, where are they? Who are they? And you can find people like this. And we've had researchers that have found each other, some validating things like, oh, I already work with them. Oh, it's so good to know that the machine learning thinks I should be. But then also some new people to collaborate with. And then with any of these little facets, if you click on the award, then you can see information like you see in the middle of the page here about the award. You can also do a search. For those who like Boolean, one of my favorite mechanisms I learned, I think, when I was 8. So you can use some Boolean and you can do a query. You can do epidemiology and lungs or all sorts of things. And then when you analyze it, it'll give you a skinny down version of the information in the database. So if we said epidemiology, right now there would be about 367 awards that would come up. And so it helps you hone in a little bit on a topical basis. So we encourage you to use this. As we say, it's your tax dollars at work, funded by the government. The COVID Information Commons Extension Award actually goes through September of 2025. So we have more time to work on this together. And we encourage researcher collaboration. We encourage student collaboration. And I think we're going to talk a little bit about the working groups. And they're working on things like long COVID. And they'll probably have it longer than we will, or they'll know about it longer than we will and study it because they may be on this planet longer. So we're very excited that we can provide this trusted corpus of information. And going into the future, one of our next steps is to have a broader metadata and data search and discovery mechanism to go beyond the information we have in the KIC and go across the internet, and perhaps also add some awards from other institutions that are vetted. So that's what we've been up to, but we're all here for you. We're here to enable researcher collaboration, help you find information that can be valuable as we all deal with the overall societal impacts of the COVID pandemic. So thank you for being part of this. I want to thank all the speakers here and the rest of the hub team that's on the call with us. Some of our students like Veralika is here, Emily, who runs our COVID Info Commons Student Paper Challenge, and a number of others. So thank you for being here, and we're glad we're in this together.
Thank you so much, Florence. And that was a perfect segue, because now I'd actually like to introduce my colleague, Emily, who's going to be speaking about another program that we want to highlight in today's talk. This is our 2023 KIC Student Paper Challenge.
Yes, thank you, Lauren, and thank you, Florence. So if you are interested in the pandemic, COVID recovery, health insights, or future innovations and discoveries, then the 2023 KIC Student Paper Challenge is for you. We welcome you to synthesize research and display your methodologies and your findings in a 4 to 5 page paper format. This challenge is open to community college students, undergraduates, graduates, and recently graduated students within the past 12 months from around the world. We also invite STEM and healthcare researchers, professors, or professionals to participate in this challenge as mentors and judges. Submissions for this challenge, which will be evaluated in an undergraduate and community college cohort and a graduate cohort for fair judging, are due July 31st, 2023. And through July 31st, participants will have access to virtual mentorship calls to assist with their research development. The very first mentorship call is scheduled for tomorrow, April 25th at 10 o'clock a.m. Eastern Time. So if you'd like to join and learn more about the challenge during tomorrow's mentoring session, please use the link in the chat to register.
Thank you so much, Emily. And I'm going to make just one final announcement before we head into today's presentations. We actually have a dedicated student working group at the KIC, and this is a project spearheaded by 2 of our work study students, Veralika and Sanket. Veralika happens to be here this afternoon. And the student working group meets once or twice per month. We have members from all around the world. Some are based in the US, others are in Europe, Africa, and Asia. So it's a really wonderful international collaboration space for STEM students interested in working together on data science projects related to COVID-19 and pandemic research. Our next upcoming meeting is actually this Friday, April 28th at 11 a.m. Eastern. So we hope that we'll see some of you or your students in that upcoming session. You can find more information about the working group, including links to our past meeting notes, details about joining the listserv, a direct link to join our Slack channel on the KIC website, and I can drop these links in the chat in a moment. So as you can see, we have a lot of really fantastic projects happening all across the KIC, and we're excited to offer these new collaboration opportunities to you all as researchers in this space. But without further ado, I'm going to now introduce the speakers of today's webinar. These are the stars of our show. This afternoon, we'll be hearing from 5 fantastic researchers. We have with us today Alan Foxman at Yale. We have Ioannis Paschalidis, who's at Boston University, Jeffrey Townsend of Yale, Chang-Yu Wu of University of Florida, and Niema Moshiri of the University of California, San Diego. And I'll kick off this afternoon's webinar by introducing you to our first speaker, Dr. Ellen Foxman, an associate professor of laboratory medicine and immunobiology at the Yale School of Medicine. She'll be sharing with us her research for screening respiratory viruses. So if you have any questions for Ellen or any of our speakers during their presentations, again, please feel free to drop them either into the chat or hang on to those questions for our live Q&A session at the conclusion of all 5 speakers. But I'm going to stop sharing my screen and let Ellen kick off our presentations for this afternoon.
All right, thank you so much. I hope, am I good? Can everyone see the screen? Okay, great. All right, well, thank you so much for having us all. It's fantastic I've been seeing these KIC emails and watching other folks present, but it's really a pleasure to have a chance to present in the KIC and be involved and reach out to this community. Today, I'd like to share some recent work from our group on host response-based screening for respiratory viruses. And I'll just start with the most important thing, which is acknowledging everyone involved who made this project happen, which includes our funding from NIH and other sources, as well as I want to acknowledge Reddy Chimarla and Amelia Hanron, who did the screens I'm going to be talking about today, as well as numerous colleagues at Yale School of Medicine and Yale School of Public Health who all worked together to allow this project to happen. So the goal of this project was to really address the issue of surveillance for SARS-CoV-2. Well, actually surveillance for new emerging respiratory viruses. There's a new imperative and high priority to do this because of the COVID pandemic. So as you all know, there's a lot of strategies already in place to look for emerging respiratory viruses. And this is really important because if we can find a virus early, we can develop diagnostic tests, we can develop antivirals and ultimately vaccines, and hopefully reduce the impact of future viruses so it won't be as impactful as this past few years of the pandemic has been. This slide just highlights some of the current strategies. One is to look in zoonotic infections to see whether there's any viruses that have a good chance of jumping into humans, and that's very important work, as well as surveillance projects for outbreaks of pneumonia, like the surveillance project that led to the discovery of SARS-CoV-2 in China, noting that there was a cluster of cases that of pneumonia with no virological diagnosis. So these strategies are very valuable, but what I'm going to talk about today is an additional complementary strategy that leverages some untapped resources for COVID for emerging virus surveillance. So this is really based on the idea of using routine clinical specimens that we get in the course of patient care to do viral surveillance. A lot of tertiary care hospitals, like our hospital at Yale New Haven Healthcare, have a multiplex PCR panel that we use for patient diagnostics. So what this is a panel with PCR tests for many different respiratory viruses, like the 10 or 15 most common respiratory viruses. Now, this isn't what you're going to get if you go in the emergency room and you clearly probably have flu or COVID. You'll probably get a rapid test or a fast PCR test for that one virus. This test is really deployed for more complicated cases where it's not clear what's going on. For example, somebody with fever, cough, shortness of breath that has sort of non-specific symptoms. And you're not sure what the clinical picture is. So even at the peak of respiratory season, when people like that get this test in our healthcare system, only about a third of them turn out to be positive for viruses, and about 2/3 of them aren't. And for the majority of those 2/3, they probably do not have a viral infection. They probably have something else causing their shortness of breath or their fever or so on. But hiding it here amongst the people where no virus was detected are probably a few cases of people that have a virus, but they just have a virus that we can't test for on our PCR panel because the virus isn't on there. The reagents aren't on there to detect that virus. And it would be great to be able to find these because some of these represent clearly viruses that we're really not thinking of as causing disease in our patients, or viruses that we don't even know about yet because they're emerging. But to analyze all of these negative samples for undiagnosed viruses would be time consuming and costly and really prohibitive because we have hundreds of these per week. So there's no way you could do deep virus discovery on each of these. It would just be, not practical. So what we sought here is a way to screen these to enrich for samples that are most likely to contain an undiagnosed virus. So you can sort of look for that needle in the haystack by just focusing in on the most important samples. And so the way that we chose to do this was to take advantage of the body's response to infection. So when the airway tissue first becomes infected by a virus, it immediately initiates an antiviral response, which involves secreting some antiviral proteins. And in prior work from our group, we showed that some of these proteins, for example, I'll talk a lot about CXCL10 today, also known as IP10 or interferon-induced protein 10. This protein is essentially one of the top secreted proteins by nasal epithelial cells when they get exposed to a virus. And we previously showed that levels of this protein being elevated correlates highly with having a significant viral load in the nose of one of many different viruses, as you see here. So we sought to use this protein as a screen and say, okay, here we have all these people with no virus detected. But among those people, who of those people are actually having an antiviral immune response in their respiratory tract? And the way that we detect that is by using CXCL10 as a biomarker of antiviral defense in the upper respiratory tract. And if we can screen for that, we reason that these people with that biomarker are going to be the high yield samples because those are the people who are likely to have an undiagnosed infection. So we did two different screens. First, I'll tell you about the screen that Amelia did in January on samples from just one single week of January 2017. So this is a week where there's a lot of viruses circulating. That week, we tested 359 samples on the respiratory panel. And the majority of them, as I mentioned, are negative for all the viruses on the panel, which at that time was only 10 viruses. Now we have more. And then we screened them for this biomarker, CXCL10, and that resulted in out of these 251, only 60 samples had the antiviral response activated at the nose. So we focused on those. But before doing virus discovery, we thought, well, we only had 10 viruses on the panel. We're missing some important ones like seasonal respiratory viruses. So what if we test for those? So we did that, and we found that actually half of these 60 samples were positive for seasonal coronaviruses, which sort of was a nice proof of concept of the strategy and also helped us focus in on the true unknowns, which represented 32 samples. So we were able to whittle down all those negative tests to only 32 samples that were of the greatest interest. Next, we chose to do metagenomic sequencing to look at all the nucleic acid in the samples and ask, are there any sequences from viruses there? And actually, we only focused on the top 10 by biomarker level because we showed previously the biomarker level, if it's higher, you're more likely to have a virus. And so we did this RNA sequencing, and we found that one of the samples, sample A, had tens of thousands of reads in the RNA in that sample that were from influenza C virus. So we felt like this is a pretty good proof of concept, because influenza C virus is a known viral pathogen, but it's not so common. And we don't usually test for it. It's not on our panel, but yet we were able to pick it up through this strategy. And to prove that this virus was in the sample, we inoculated primary human nasal epithelial cells with the actual patient sample, and we were able to observe cytopathic effect from the virus, syncytia formation, as well as that virus in the secretions of these nasal epithelial cells 7 days later. So we found, our first pass at this strategy worked pretty well in identifying a sample that had an undiagnosed clinically significant virus. The next screen I'll tell you about was, we were actually sort of wrapping up that project when COVID came along. And we thought, you know what, I wonder if we could use this same strategy to find undiagnosed cases of SARS-CoV-2 from the beginning of the pandemic. If you look at this graph here, the gray represents case counts in the United States in March of 2020. And then blue is New York State, which is pretty close to our hospital. A green is Connecticut, and then the red is our hospital. And the bracket here shows this time between all these viruses entering the New York region and the time when our PCR test was up and running for SARS-CoV-2. And in those 2 weeks, we tested 375 samples on our respiratory panel that all tested negative. So actually, it was about half or 60% of the samples from that period tested negative. And we thought, well, let's see if this biomarker would pick up any undiagnosed cases of SARS-CoV-2. So we screened with the biomarker, and in parallel, we screened with by RT-qPCR. There's the typical PCR test for COVID. And what we ultimately found was that there was 4 undiagnosed cases of COVID that we discovered by PCR, as shown in these red dots here. And then this graph represents the level of the biomarker. And what you can see is, for the majority of samples, for 92% of the samples, the biomarker was below our cutoff threshold. But for a small percentage, about 8% of the samples, the biomarker was above that threshold. And all of the COVID positive samples were in that group. So if we had wanted to, we could have done the biomarker screen first and only tested 1/10 of the samples by PCR and still found all the undiagnosed cases. So this was a really good way of getting a comprehensive view of how this strategy could work. In addition, the advantage of using these nasal samples for the screening is we were able to directly, with our colleagues from the Gruba lab, get the sequence of those viruses and do molecular epidemiology, which is their specialty in Nate Gruba's lab. And so what they found was that, interestingly, all these 4 isolates from that early time point in the pandemic were all genetically distinct. So that represents that SARS-CoV-2 was coming into our healthcare system and into Connecticut by multiple different lines of transmission at that time. It wasn't all from one source. So that kind of gives you the bigger picture that the transmission was much more widespread than we realized by that time. Finally, don't have time to really discuss this today, but I just wanted to point you to the paper if you're interested, and let you know that we did a lot more deep characterization of these unknown samples and compared them to known cases of virus positive and virus negative patients. And we looked at the nasal host response to infection using RNA sequencing and cytokine profiling. And you can see more subtle patterns. So mostly today I talked about elevation in one biomarker, CXCL10, but there are actually numerous patterns that you can pick out that might be helpful in the future for developing more sophisticated host response screens and learning more about undiagnosed infections in the patient population. So just to summarize, the key findings of this study were that we used a single biomarker of the nasal mucosal response to viruses to enrich for samples containing undiagnosed viruses. So we used these patient samples testing virus negative and used the biomarker to highlight which samples we should further pursue. And these screens diagnosed an undiagnosed case of influenza C virus, as well as 4 undiagnosed cases of SARS-CoV-2 that were epidemiologically significant. And what we're doing now is we would like to scale up this screening by making the workflows more automated, and there's a bit about that in the paper. We're also interested in collaborating with people who might have samples from different geographic regions, because obviously in different regions, you may have different undiagnosed viruses circulating. And we're also interested in using these nasal immunophenotyping to help further parse what undiagnosed infections are going around in our patient populations. So with that, I'll conclude, and I'd be happy to take any additional questions at the end of everyone's talk. So thank you very much for your attention.
Thank you so much, Ellen. That was really fascinating. And I'm sure we're going to have a lot of questions for everyone at the end of the presentations this afternoon. So I'd like to introduce our second speaker, Ioannis Paschalidis, who's the Distinguished Professor of Engineering and the Director of the Hariri Institute for Computing and Computational Science and Engineering. That's a mouthful at Brown University. Ioannis, please feel free to kick off your talk. Thank you.
Thank you so much, and thank you for inviting me to be part of this exciting afternoon with a great set of speakers. So I will talk about predictive models of COVID-19 severity and patient outcomes that we have developed at Boston University. And just to give a little bit of a context, we've been working for quite some time before the pandemic on a variety of models that predict disease and important key events, for instance, hospitalizations, and also on models that prescribe treatments. So when the pandemic started, we mobilized, like the rest of the scientific community, and we also mobilized a relatively large network of collaborators. So we obtained access to a variety of different data sets, and you can sort of see in this slide the various data sets. We had data sets that were local from Massachusetts, from two different hospital networks in Massachusetts, one from the Mass General Brigham network from 5 different hospitals, about 2,500 cases, and then another relatively large cohort from the Boston Medical Center, about 7,000 cases. We also got some cases from Wuhan, China, which was the origin, obviously, of the epidemic. And finally, we're able to get access to some large national data sets, so a data set from Brazil and another data set from Mexico. So in this very short presentation, I will focus more on the most recent work that considered the data set, the series of patients from the Boston Medical Center. The Boston Medical Center is the teaching hospital affiliated with the BU Medical School, and is also a safety net hospital. And as you will see, this has some interesting implications in the findings that we were able to get. So we got access to the entire 2020 BMC cohort, about 7,000 patients. So those were patients who tested positive for COVID-19. And just some rough statistics, about 20% were admitted. From those admitted, about 23% or so were admitted to the ICU. From those admitted to the ICU, about 58.7% were intubated. And from those intubated, about 70% unfortunately did not make it. And we had lots of information about these patients, including demographics, their vitals throughout their hospital stay, radiology reports, their medical history, any symptoms, any lab results, any medications, and even information about depression status, zip code, and also we had information about the occupancy of the hospital at the time that each one of these patients were seen. We also had information on social determinants of health. BMC runs a program called the Thrive program, that everyone who has an encounter with the hospital is being given a survey, and that survey asks them about need in a variety of different areas, including housing, food, transportation, help with caretaking, medications, help in getting access to medications and paying for medications, education, and employment. And a lot of the information was in tabular format that is more easily handled by AI machine learning methods, but also there was quite a bit of information, particularly radiology reports and other reports during the hospital stay that were just narratives, reports by clinicians. So we had to use quite a bit of natural language processing in order to extract appropriate findings from the text. And what we did is we developed a set of robust interpretable models that can predict hospitalization, ICU admission, mechanical ventilation, and death. And I'll show you some examples. I will not be exhaustive, again, in the interest of time. So first, what we did is for every patient, we constructed a timeline from the time that we were aware of the patient testing positive and having information about this patient, and up to the point of the event of interest, whether that was, let's say, an ICU admission, whether that was a mechanical ventilation. So here is the outcome of interest. And then looking back in time, we created these data buckets. We dropped any information that was available just before the time of the event of interest. That is for reasons we wanted models that could predict what will happen into the future. Perhaps even a first-year medical student can identify a patient heading to the ICU. So we wanted the models to make that prediction with earlier information. And then you will see that we obtained different versions of the models that had different cutoffs in terms of the information that the model used in order to make the prediction. Another reason we created these time buckets is we wanted to capture the dynamic evolution of the progress of the patient while in the hospital. For instance, the dynamic evolution of the vitals, because we understood that this was rather important. So rather than just looking at a snapshot of vitals, let's say at some specific time, and using that information for making a forward prediction, the exact values are important, but also is trends are important as well. And physicians, when they look at patients, they sort of look at the trends of the vitals in the patient. So we developed a model that used some fairly sophisticated deep learning methodologies, including LSTM type of networks and a transformer architecture that took as input the vitals, 6 vital signals at different points in time, and produced a score. And that score captured the dynamic evolution of the vitals, and that vital score was then used in an ensemble model that was attempting to make a prediction for the outcome of interest. So, for instance, hospitalization predictions, you can see that these are fairly accurate. So some of the best models give you a 92%. This is in area under the curve. You can think of this as a measure of accuracy of the model. The best is 100%. A random guess will give you 50%, so 92% is quite good performance. And you can see here that from some linear models that we developed, we also found some of the factors that were important in making the hospitalization prediction. And in blue, you see some of the variables that are associated with some earlier health conditions that, for instance, are highly correlated with hospitalization. You will also see that the occupancy of the hospital, if it was high, was reducing the likelihood that the patient is going to be hospitalized. And also you will see 2 social determinants of health, need for food and need for transportation, that were both contributing to a hospitalization decision. So patients with those needs were more likely to be hospitalized. And I would like to emphasize the role of these social determinants of health. This is something that we also saw in other data sets, particularly in the Brazilian data set. That was a national data set, and we found that socio-demographic factors were impacting hospitalization decisions. What we also found was that the model, the naive model that one is able to produce is actually rather biased. So you could see here how the model performs out of sample for Black individuals and white individuals. And the false positive rate of the model for Black individuals was twice as much as the model as the false positive rate for white individuals. Even though we were controlling for race, we were controlling for socio-demographic factors, we were controlling for social determinants of health. Despite that, the model was much more eager to make the prediction that a Black individual was going to be hospitalized compared to a white individual, and correspondingly, it was most likely to make a false negative prediction for a white individual compared to a Black individual, which suggests that there are apparently hidden features in the data that are not visible to us, perhaps reflecting structural bias and other factors that make the model make that biased prediction. And there are ways, and we have addressed them in a paper we published in JEMIA, on how one can correct for these factors and produce models that do not have this sort of bias. So these are some results on ICU prediction, predicting an ICU admission. Roughly the median gap between an admission to the hospital and admission to the ICU, at least in our data set, was about 4 hours. So we will use different cutoffs. If you use the latest information, then you get quite accurate models with AUCs on the order of 93, 95%. And if you start cutting off the information that you were going to use, so 12 hours in advance, the performance of the model drops to about 86%, and 24 hours in advance, the performance of the model drops to about roughly 80%. And what I would like to emphasize is that we compared these models that we developed to some standard models that are predicting ICU admission. So there are some well-known sepsis models, NEWS, SOFA are called, and these are fairly inaccurate in this case, indicating that standard models for ICU prediction, at least in the COVID cases, fail to predict an ICU admission, indicating the rather unique signature of the disease. And here you find some of the variables that were, again, highly correlated with the outcome with an ICU admission. And what was interesting to us was that this vital score that we produced that captured the dynamic evolution of the vitals pretty much tells the entire story. And there are some other variables or some lab variables, LDH, CRP, that have been identified by other studies that also are contributing. But if one just takes the dynamic evolution of the vitals, that pretty much tells the ICU admission story. Finally, we produced a number of calculators that we made available on the web. These, I understand, were used by our colleagues, collaborators at Mass General Hospital in the early stages of the epidemic. It was very easy to input some of the key variables and get a prediction, for instance, for an ICU admission or for a mechanical ventilation need of a patient. And we found that, we were challenged to find cases. What is the model telling me that a very experienced clinician cannot potentially predict by just looking at the patient? And here are some cases, and there are many others, where patients were admitted, they were stable for a couple of days. Nothing in their clinical outlook suggested that the condition of this patient was going to deteriorate, but then the model was able, upon admission, based on some special laboratory results predict that the patient was going to need ICU care. So these are two cases with the details of those cases. So that brings me to conclusion. Of course, I'm just presenting. There have been many people that have contributed to this work, and I would like to thank them, including students in my group, but also our collaborators at Mass General, Brigham, Boston Medical Center, and some of the other areas where we've been able to get data from. So thank you so much for your attention, and looking forward to questions at the end of the session.
Thank you so much, Ioannis. And again, given the insights we've heard so far, I think we're going to have a very active Q&A session. So let me introduce our next speaker, Jeffrey Townsend, also at Yale, along with Ellen. He is the Elihu Professor of Biostatistics at Yale School of Public Health and the Professor of Ecology and Evolutionary Biology there. This is actually Jeffrey's second KIC presentation. We first heard about his research on zoonoses in a 2021 webinar. So this is a long-awaited research update. So Jeffrey, the floor is yours.
There we go. Thank you for the introduction, and thanks to both of the folks who preceded me for presenting a lot of interesting information about COVID-19 that I fortunately don't have to talk about. So I'm going to talk about the durability of SARS-CoV-2 vaccine-mediated immunity and the optimal timing of booster vaccination, which of course is relevant to all of us now as we think about this disease. And I just want to point out some of the people I collaborated with, particularly Haley Hassler, who's listed second here, who was a research assistant for this project, but a very hands-on, very highly involved research assistant. She deserves enormous credit for this, and also was the original author of the slide deck I'm going to present. So a lot of credit to her. And then Alex Dornberg, who's listed last year, who was a collaborator with me on the whole project from the very beginning, and with whom I've enjoyed working on many projects. So there have been innumerable questions regarding the COVID-19 pandemic. There have been questions, can computer models be the key to better COVID vaccines? Could a universal COVID-19 vaccine defeat every variant? How often can I be infected with the coronavirus? Can you get a coronavirus from drinking Corona beer? No, but the many, many questions have arisen. One of the major ones that was very pressing early on was the question of how long does immunity to COVID-19 last? And research to try to give a result to this was sort of waiting for data to appear, because the problem with immunity is it's something you see over time, and without time, you don't have data. So one question, if I've already had COVID-19, will I get it again? We all are familiar with the sort of itinerary of COVID-19 and how it has spread through communities and then spread through a second time, even a third time in some cases. Another question, if I'm fully vaccinated, am I immune to COVID-19? And we've had a lot of people vaccinated around the globe, around 62% right now, and some people many times boosted, some people a few times boosted. So there's a lot of variation there. Would we need to keep getting COVID-19 boosters forever? And how will we answer that? So we have previously provided estimates, as was mentioned, to answer the first question, which is if you've already had COVID-19, will you get it again? The answer is largely yes, unless there's some other preventive method being used. And that result came out in a paper, The Durability of Immunity Against Reinfection by SARS-CoV-2, which was a comparative evolutionary study, very interestingly, because as I said, until data accumulates over a very long period of time, you can't really accurately quantify how much immunity people get to a disease. But we do have an enormous amount of data over time accumulated for a variety of coronaviruses, the seasonal coronaviruses that Ellen Foxman mentioned earlier. And that data allows us to understand things about SARS-CoV-2 because they're closely related, and evolution is not something that dramatically changes such things over relatively short periods of time. These are not hugely diverged viruses. All right, so what did we do to actually analyze these kinds of questions? We wanted to make estimates of how how frequently people got reinfected. And a very important data set was collected by Edridge et al., which is portrayed, these are just 3 samples from that data set, looking at longitudinally at the antibody levels for seasonal and endemic coronaviruses. So we could take those seasonal endemic data for the known coronaviruses that have been around a long time, HCOV 229E, NL63, and OC43. We could also take antibody information that we know about the decline of antibodies after infection from MERS-CoV and SARS-CoV-1, and put all of that data with the little bit of data over a short period of time that's already been collected for SARS-CoV-2, and then place it on an evolutionary tree. That is, look at how each of these different pieces of information about how declines happen after infections for the seasonal coronaviruses, all 3 of them here, the MERS-CoV for SARS-CoV-2 and SARS-CoV-2, and actually look at how this decline in antibody levels happens for different diseases based on the viral divergence between them. That enables us to fill in sort of not just the original short period of time that we have data on, but the entire trajectory anticipated for antibody decline for SARS-CoV-2. And more than just the antibody decline, it also allows us to figure out whether individuals are likely to get infected or not, at what level of antibody that they have, because we can infer that, again, based on the seasonal coronaviruses for these other infective coronaviruses, especially SARS-CoV-2, which is now approaching endemicity here. And one of the things I just want to emphasize is that our estimates from that analysis were actually validated by empirical studies. So as time went on, it became easier and easier to track how many people got reinfected after getting first infections. And in one study by Malhotra et al., 34% were of unvaccinated individuals were reinfected at 420 to 480 days. Our estimate from the evolutionary approach was that 34% probability of infection would occur at 450 days. And another estimate was that at 82% of people got infected at 275 to 300 days. And our estimate was 82% of no reinfection at 270 days. So these were very, very consistent estimates from empirical data for the projections that we made, a year and a half before, based on comparative evolutionary analysis. So it's kind of analysis that works. It tells you the correct answers. So our research addresses 2 additional questions here. If you're fully vaccinated, are you immune to COVID-19? Not so much. Will you need to keep getting COVID-19 boosters forever? So to answer that question, we looked at 4 different vaccines, the 4 that are most likely prescribed, that have the greatest global reach, which is Pfizer, Moderna, Oxford, AstraZeneca, and the J&J vaccine. And I just want to emphasize that the question is, over 6 years, would you need to get that Pfizer vaccination, or would you not, and how frequently? So comparing the peak in antibody response of natural infection to that of the Pfizer-BioNTech allows us to project to the actual vaccinations what we expect the level of antibody how much prevention of infection we expect antibodies to confer. And we can look at the antibody levels over time for the different vaccines, and we see they have different curves. And the point is that we get shorter immunity when this curve is lower here, when the peak normalized antibody levels are lower, and we get longer immunity when it's longer. And we can also look by using that information about what antibody level people get infected for these different diseases, we can get a probability of no breakthrough infection over time. This is how many days after the original infection someone had, and what the likelihood of infection will be. And there's one thing that's not on my slides I just want to emphasize here. This accounts for the evolution of the organism over time, because the seasonal coronavirus data included that evolutionary phenomenon as well. Seasonal coronaviruses, just like SARS-CoV-2, evolve. Getting infected by them once doesn't make you immune for the rest of your life. Same phenomenon. All right, so as I said, that longer and shorter is conveyed by that information. So we compared peak antibody response to natural infection to that of Pfizer-BioNTech. So we needed a way to normalize all of them. What we did was we normalized all of them to the Pfizer-BioNTech data, which had the strongest data set comparing unvaccinated reinfection and vaccinated reinfection, et cetera, et cetera. And we find that the peak level This is just a relative level of antibodies after for the Pfizer-BioNTech vaccination is 1.5 compared to natural infection at 1. And again, you can draw this out over time as to what the antibody levels are over time. Now this is just a big plot to show you. We used a lot of data. We compared the Pfizer data to that of the other 3 vaccines. This is work, again, Haley Hassler did all of the extraction and work to do this. Essentially, looking at that normalized Pfizer data to natural infection, we then related the other 3 vaccines to BNT, where we had comparative results within the study using exactly the same markers, exactly the same technology, et cetera, within the lab to assess antibody level. Because if you use different technologies, you get different numbers. And the point is, all of these studies showed Pfizer plus Moderna. All of these studies showed Pfizer plus Oxford and AstraZeneca, and these studies showed Pfizer and J&J. All of that data coming together enabled us to figure out information, and we made sure for all that data that it was sampled near the peak antibody response, that they were COVID-19 naive subjects, that there was a proper vaccination timing being used, and that there was that, as I said, comparison to Pfizer-BioNTech. All right, given that, we found that antibody levels for mRNA vaccines were higher than natural infection, while viral vector vaccines were approximately similar to natural infection. So here are the plots for that. The red line is for Moderna and Pfizer, quite similar to each other in response, in terms of antibody response and decline. And the blue and teal lines, well, the teal line is J&J, is Oxford AstraZeneca, the blue line is J&J. And what that tells you is that breakthrough infections for those vaccinated by either mRNA vaccine were predicted to typically occur over a longer period than natural infections, or breakthrough infections following either viral vector vaccination. So here are the plots for that, just conveying the number of days you're expected to get a breakthrough vaccination after vaccination with mRNAs, natural infection, and those 2 viral vector vaccines. So just to summarize it very simply, the mean time by which there's a 5% cumulative risk of breakthrough infection for mRNA vaccines is more than twice than that of natural infection or viral vector vaccines. So greater efficacy in protecting you over a longer duration from the mRNA vaccines. Just going to skip that to point out, we compared the peak antibody response of primary vaccination to that of the boosting in order to assess what boosting did. What you see is you get a higher dose after boosting. We're very careful about the timing involved here as well. But the point is we have a peak of about 1.54 when you get brought up by the boost in terms of your antibodies. And that allows us to do the same question. I'm not going to go into the details about this, but just asking over time, how frequently, what frequency of vaccination is going to give you what level of resistance to future infection by COVID-19. I just have this slide and one more. And the point is that the cumulative probability of getting a breakthrough infection, of course, increases over time. This is over a 6-month period. And the point is, if you have no boosters, you have about an 87% chance of getting infected over a six-year period. This is with endemic, not pandemic disease. So this is where we're headed, not where we are. Over, if you get 1 booster every 3 years, it does not very much to reduce your chance of getting infected from 87 to 77%. Over 2 years, drops it down to 64%. Every 1.5 years, drops it down to 51%. And then we really start getting somewhere with every year vaccination, that's down to about 31%, 1 out of 3 people. If you get a vaccination every 6 months, these are in healthy people, by the way, that's every 11%. That's only 11% over 6 years. So very low chance, or relatively low chance over an extended period of time. So it has a tremendous benefit in terms of prevention of infection. And I just want to emphasize that this is the result for the Pfizer-BioNTech. The result, if I can get my slide to switch, for mRNA is very, very similar. So for the Moderna vaccine is very similar. We weren't able to do it for the other 2 because the data doesn't exist in the same format. But that's the general result for those mRNA vaccines. And with that, I'll just finish with my conclusions. the mRNA vaccines provide immunity over a longer duration compared to natural infection or viral vector vaccines. Delayed administration of boosters is of high consequence to the cumulative probability of SARS-CoV-2 breakthrough infection. So if we want to suppress it, administration of boosters on as much as a 6-month scale would suppress the disease spread considerably, especially when you think about that as those results I showed you are the results for an individual when it is spread at an endemic level. So they aren't accounting for the herd immunity provided by the vaccination itself. That's just supposing it stays at an endemic level and you get vaccination, how likely are you to get infected? Last point, population-wide booster vaccination can forestall and potentially strongly suppress COVID-19. One aspect that maybe I just want to emphasize is that I think we all got a little bit jaded on the boosters in some sense because the booster kept being given that was to this ancestral strain. It makes a big difference to have these updated boosters. And I think that has not been widely understood by the public and actually even many in the epidemiological community. Thanks to all my collaborators who helped me do this.
Thank you so much, Jeffrey. That was really interesting. I'm going to now hand it over to our next speaker for this afternoon, Chengyu Wu, who is the chair of the Department of Chemical, Environmental, and Materials Engineering at the University of Miami. Dr. Wu will share his research on aerosol transmission of COVID-19. Chengyu, it's all yours.
Thank you. Can you hear me? Yes. Okay, good. Yeah, thank you for the invitation. We are very happy to share our research endeavor in assessing aerosol transmission pathways of this virus. Okay, so going back to the beginning of the pandemic, it was a very chaotic time because we had no clear idea how the virus was transmitted from one to the other. And based on the previous knowledge about the flu, transmission. So WHO and the CDC advised that, oh, these are probably by droplets, or they may be transmitted by contact, like fomite transmission. So in that case, if you can maintain social or physical distancing, or you wash your hands, you should be fine. That was in the beginning of the pandemic. But as an aerosol scientist, I had a question about that because respiratory virus is supposed to be airborne. But let's just wait for 1 minute. So if you follow the physical distancing and the washing hands, are you protected? Well, apparently there were quite a few cases that told us not really. So one example is in the Skagit County in Washington State. So following a 2.5-hour acquired practice, attended by 61 people, 45 were infected and 2 people died from that. And they practice the physical distancing and also they wash their hands. So definitely it's something else. So at that time we said, we have to do something. We have to do air sampling to prove that it is transmitted in the airborne state. So how do you do that? Conventionally, you would use this kind of air samplers, and which are filter based to collect these particles on filter, then to do the analysis. However, these viruses may get inactivated due to desiccation during sampling. And also the other challenge is the recovery of virus from filter may be an issue for certain filter. So, and if the viruses are not viable anymore, then we cannot really convince the WHO doctors that, oh, the airborne transmission is an important pathway. So how about collecting them in the liquid medium using these type of samplers that can help conserve their viability? That's a good idea from the conservation perspective. But if you look at this figure over here, and you can see the collection efficiency is very, very low. It's at 5 to 10% for the 100 nanometer particles. And we know that the SARS-CoV-2 variant is about 100 nanometer particles. So we are in a dilemma. How can we efficiently collect the virus aerosols and maintain their viability? That was our challenge. So we developed a device inspired by nature process. Okay, so this happens in our respiratory system. When you have these virus aerosol getting into the human respiratory system, and then there will be water vapor condensing onto these particles and making them much larger. So in that case, you will be able to collect them more efficiently. So we actually engineered the same device using this same principle in this device, which we call the viable virus aerosol sampler. So first you cool the particles into a cold state, and then next you introduce them into a moist environment. You have a lot of water vapor condensing onto these particles, making them much larger, and at the same time conserve their viability. So you can collect them and do analysis. And this is the photo of our device. Okay, so how good was it? So we first tested with lab-generated H1N1 virus. The x-axis is the amount of infectious virus generated during the sampling, and the y-axis is the number of viruses, infectious viruses collected. And as you can see, the virus was very close to the 1-to-1 line, which is the ideal situation. And the biosampler, which is the industrial standard, and was one order of magnitude lower. So this test demonstrates the superior performance of the virus compared to the biosampler. Okay, then it was the pandemic time. And so we took our virus into a hospital housing the COVID patient. And at that time, WHO said, the physical distancing, if it's more than 2 meters, you will be safe. And we wanted to prove that we have to be careful about that. So we placed our samplers, and this BioSpot is the commercial version of the virus. So we put the two samplers at 2 meters away from the patients, and we collect the air samples, and we compare with the human specimen from the patient, and they match. So this demonstrates that the aerosol can be a possible route for the transmission of the virus. And furthermore, we inoculated the cells with these air samples, and you can see that after 4 days, 7 days, and 10 days, the cytopathic effects of the cells. So they are infected and died because of the infection of the virus in the air sample. And also, as days goes on, you can see the CQ value decrease. That means a higher and a higher concentration in the sample. That tells us that the viruses are growing in the cells. So that means the viruses are viable. So this was the first study to show viable SARS-CoV-2 in the air greater than 2 meters away from the COVID patient. And New York Times reported this is a smoking gun to what WHO and the CDC advises how we can better protect ourselves. We have to consider really the airborne virus, not just the droplets or the fomite transmission. And so this finding provided evidence that helped change of the WHO and CDC guidelines. Okay, but the virus transmission doesn't just happen in the hospitals only, especially after there were a good practice of using the personal protective equipment. So we were thinking, where else does this transmission happen? Where is the hotspot? And our hypothesis was that actually the residential space would be the hotspot, because at home, typically you don't wear masks and there is no social distancing. And further, there is no constant ventilation to reduce the concentration of the virus. So we did one sampling in a volunteer's house, and this is where the infected individual sits. So this is the isolation bedroom. But we also conducted sample in a bedroom far away from the isolation room, which we call the bedroom 2, and in the same household, but at different rooms. And you can see the collection of the samples in the isolation room and the bedroom where the person was supposed not to be there. And we were able to culture the virus in the air sample at the isolation room. So this was the first study showing viable virus in air samples outside of the healthcare facilities and on the phone. And this is because of the use of this new tool. And so what we learned from here is that SARS-CoV-2 aerosol can be transported to other rooms far away in the same building. So that will change how you would deal with what kind of advice you would give to people to better protect them from the exposure. We did some more sampling in the in several volunteers' room. And here we have the primary room and the secondary room. Primary room is the self-isolation room where the infected individual spend most of the time. And the secondary room is the outside of that room where the infected individual doesn't really spend too much time, at least according to what they told us. And the the y-axis is the concentration of the viable virus. And you can see essentially statistically, there is no real significant difference between these two. What does it mean? Well, this tells us that the the risk in the primary room and the secondary room, it probably is very similar. And so this was the first study showing the air sample in the secondary room, and the viable SARS-CoV-2 can be transported to other space in the same building. Again, this is due to the use of the new tool that was developed. So what is the application or implication? Several of them. The new knowledge that we learned from the sampling tells us that, hey, you got to have a good ventilation when there is someone sick in the residence, right? And there are also other things you should do to protect yourself, like wearing a mask when there is a co-occupant that is sick, who is sick in the space. And also, very importantly, we demonstrate that you need to use the right air sampler in order to give you the correct information, because there have been a lot of studies using the conventional air samplers. Then they are not able to capture the virus, and they will say, oh, it's fine, it's not really an issue, but that is because of the limitation of the samplers that you used. Okay, now I'm going to change the topic to a somewhat different direction, which is also part of our project. So the virus was very good in collecting the samples, but the analysis takes days. And very often we would like to have the information, is the virus over there in the space in a short period of time, right? So that's the point of having this point of care detection. We wanted to be able to do the analysis right over there, and we will have the answer very quickly. So here we have my collaborator has developed this VD device, and this is where you have the samples from the virus. And you don't have to use pipettes in this analysis. You just need to slide this device from 1, 2, 3, 4. You can do the lysis of the samples, the RNA binding and washing. And then the mechanism is like the ballpoint pen. You have this push pen that will release the chemicals, and you will do all these things without using pipettes, and everything will be done within 1 hour. And the results shown over here that we were able to detect the SARS-CoV-2 virus or influenza virus right over there within 1 hour. So you don't have to take the samples back to your lab. Okay, so finally, just the summary. I hope I have demonstrated that we have to use the right tool, and water vapor condensation is a very good method to help us amplifying the particle size and therefore conserving the viability of the virus for infective analysis. And with this tool, we were able to collect and isolate from air samples from hospital room, the primary room, and the secondary room, far away from the primary, using this device. And so from that, we know that the good ventilation and the PPE, they are very important in keeping low risk in the indoor space. And additionally, if we would like to develop the point of care detection capability, so we can have rapid risk assessment of exposure to the respiratory viruses right over there. You don't have to take the samples back to your lab for analysis and wait for days. And finally, I would like to acknowledge the financial support from NSF and NIH, and also my collaborators and students working on this project. That's all I have, and thank you very much for your attention.
Thank you so much, Shengyu. And now I'm going to introduce our final speaker for this afternoon, Niema Moshiri, the Assistant Teaching Professor in Computer Science and Engineering at UCSD. Like Jeffrey earlier, Niema has actually presented his research at a previous KIC webinar in 2020. So this is another update on that initial presentation that we've been anxiously awaiting. So Niema, please take it away.
Awesome. Yeah, thank you for the introduction. And hopefully folks can see my screen. So hey everyone, as was mentioned, my name is Niema Moshiri. I'm an assistant teaching professor in the Computer Science and Engineering department at UC San Diego. And then my talk is going to be focused on some methods that my lab developed using the NSF funding for speeding up viral genomic analysis. So specifically, today's talk is just going to focus on how we've enabled massively scalable reference-guided multiple sequence alignment of complete viral genomes. We actually have done multiple other accelerations as well that I just didn't have time to talk about today, but I'll kind of end with a link to my website if folks are curious about how else you could speed up other aspects of this type of analysis. But yeah, so let's get started. So just to give a little bit of context, here's a framework for a standard viral phylogenetics workflow. And before I even talk about this, viral phylogenetics is very important to be able to study how is the virus mutating over time. How is it kind of branching off? How are the different samples that we collect all over the world related? There's a ton of uses in the world of viral molecular epidemiology that are out of the scope of this talk, but just generally having a phylogeny inferred from viral genomes is very useful to have. So typically the workflow starts like this, where you start with a bunch of unaligned viral genome sequences, which I'm showing over here. The first step is usually multiple sequence alignment, where you try to kind of place these gaps in the different kind of positions of each of the sequences to get them to line up better. And this gives you some notion of sequence homology after you do this. Then given the multiple sequence alignment, we can then perform phylogenetic inference to try to infer an unrooted evolutionary relationship between these sequences. And then typically after that, we do what's called rooting to determine what is the most likely common ancestor of all of the sequences. And that kind of then tells us what was the forward in time evolutionary history of these sequences. Then maybe you'll do some additional downstream analyses. So maybe you do transmission clustering. There's a lot of other analyses that you can do on the phylogeny and on the sequences. But yeah, this is kind of the building blocks of how you do all these other analyses. So typically, these steps over here are the key computational bottlenecks: the multiple sequence alignment and then the phylogenetic inference. In today's talk, I won't be talking about phylogenetic inference. I'm just going to be zooming in on multiple sequence alignment. So some context: multiple sequence alignment. This is what's called an NP-complete computational problem. What that means, there's a very technical computer science term, but basically what this means is there's no polynomial time exact solution. So basically, if you gave me a bunch of sequences and asked me to come up with the optimal multiple sequence alignment, there is no way to do this in polynomial time. It's very, very slow. Heuristics have been developed to provide approximate solutions. So for example, you might have heard of Cluster Omega, Muscle, Map. These are some kind of standard tools that are used in this space. However, even these heuristics, they generally scale quadratically with respect to the number of sequences. And for context, the GISAID database, which is the database where most folks are storing their full SARS-CoV-2 genomes, this database is growing extremely rapidly. And as of today, we have over 15 million SARS-CoV-2 sequences available from all over the world. The next epidemic is going to be more, and kind of sequencing genomes in real time. This is going to be a tool that hopefully we continue to use in the viral epidemics to come. So we can expect this to be even more significant of a big data problem. So currently with these tools like Cluster Omega, MAFT, and Muscle, we're looking at runtimes of decades to centuries, which, for obvious reasons, if we're trying to do real-time molecular analysis, decades or centuries is just a little bit too slow. So how can we speed this up? Well, it turns out that the problem is actually a little bit easier than what we're trying to solve. Multiple sequence alignment in general is kind of assuming no homology of the sequences whatsoever. This is the time it takes to align completely arbitrary sequences. But with SARS-CoV-2 and with viruses in general, we have a much simpler problem, right? We have a lot of sequence homology, and even if the virus is mutating, significantly across the world, every single viral sequence that we obtain is going to be almost identical to the reference genome. It's not going to be exactly identical, but it's going to be almost identical. So we actually are facing a much simpler computational problem, which is multiple sequence alignment of highly similar sequences. So how can we use that feature to speed up this analysis? We can do what's called an align to reference approach. So instead of just trying to align everything with each other all at once, what we could do is do individual pairwise alignments against a reference genome. So in this figure, the thicker green bar at the top represents the reference SARS-CoV-2 genome, and each of these other colored genomes represents a sequence that I collect from the real world. And I want to align each of these to the reference genome. And what I can do is just one by one by one by one, I can independently align each of these genome sequences against the reference genome, which I could do each of these fairly quickly, and I can do massive parallelization because each of these pairwise alignments to the reference can be done completely independently. So I can parallelize however many cores my computer has. I can throw that many at this problem. And then once I've computed all those pairwise alignments to the reference, I could use the reference genome. I can kind of use its anchors, its positions as anchors to create the columns of my multiple sequence alignment. So for example, maybe I'll start with the first position of the reference genome, and I'll see, okay, well, in the red sequence, this is the letter that aligned to that position. In the orange sequence, this is the letter. In the pink sequence, this is the letter. In the blue sequence, this is the letter. And I can kind of merge all of those letters into one column of my multiple sequence alignment. And then I can do the same thing for the second position of my reference genome, the same thing for the third position, fourth position, all the way across, and kind of position by position by position, I can build my multiple sequence alignment. So this is really good because it is massively parallelizable and it scales linearly with the number of sequences rather than quadratically. So much better scalability as well. So do we have to implement this approach from scratch? We actually don't. It turns out that if you kind of step back and think about this problem, this is really equivalent in a sense to the long read mapping problem. So let's just kind of step back and rethink what is the problem that we're tackling. Our input is a reference genome and a bunch of long sequences that are very similar to the reference genome. And our output is an alignment of each of those sequences against the reference genome. This is exactly the same computational problem as mapping long reads. So instead of having to reinvent the wheel, we could just kind of build off of all of these really advanced techniques that folks have built to solve the long read mapping problem and just kind of apply it to this context. So to that aim, I developed a tool called Viral MSA. And what it does is it just wraps around existing long read mappers to perform this reference guided multiple sequence alignment. So it kind of treats each of those genomes that I've collected as long reads, and it treats the reference genome as a reference genome, and it just calls that read mapper. I wrap against a few different read mappers just to demonstrate flexibility, but I mainly suggest people use Minimap2 for both speed and accuracy. And then given those read mapping results, given the mapping results, I can then just kind of compile them into a single multiple sequence alignment. So yeah, all you have to do to run Viral MSA is you just give Viral MSA a reference genome and a bunch of sequences to align, and it'll automatically handle indexing the reference genome. Maybe if you give it an accession number, it'll handle downloading and indexing the reference genome. It'll handle all of the pre-processing and all the downstream stuff. And it'll just output, it'll call the read mapper, it'll merge the results into the multiple sequence alignment, and it'll just output a single standard FASTA file that is your multiple sequence alignment. So how does it do against existing tools? Well, if we did a benchmark experiment where we compared the runtime of Viral MSA wrapping around Minimap2 compared against Viruline, which is an existing align to reference approach, but that just kind of implements its own from scratch align to reference. And we also compared against MAP, which is typically considered one of the most commonly used multiple sequence alignment tools. So in this plot on the horizontal axis, I have number of sequences. On the vertical axis, I have total execution time in seconds. And this was done on complete SARS-CoV-2 genome sequences, so genome length roughly 29,000. And as we can see, the blue line, which is Viral MSA, is orders of magnitude faster than the existing tools. Compared to Viruline, which is also scaling linearly, we're getting, and by the way, this plot is log scale plot. So compared to Viruline, we're roughly like 1,000 times faster-ish. And with MAF, we're not quite as much faster, but you can see that because MAF scales quadratically, our speed up with respect to MAF is actually increasing as time progresses. So even at just 1,000 sequences, we hit roughly 1,000 times faster, and that gap kind of increases. Now you might be wondering, okay, well, fast is good, but what's the point if it doesn't give me good alignments? Well, we also compared the accuracy. What we did was we took the multiple sequence alignment computed by MAF, took the multiple sequence alignment computed by Viral MSA on a bunch of hand-curated alignments from HIV, Ebola, and I'm blanking on the third virus. But basically we took viruses that we had curated alignments from the Los Alamos. Oh, actually, no, this plot is from just HIV-1. From the Los Alamos National Lab, we took their curated multiple sequence alignments and we used that as ground truth, and then saw how does mapped and viral MSA compare against the ground truth curated multiple sequence alignment. So if we compute pairwise distances of the sequences that we get in our alignment, and then we do a mantle test for accuracy, we find the correlation between our pairwise distances and the pairwise distances calculated directly from the true multiple sequence alignment. We see that the correlation is negligibly different. So here, we get a correlation coefficient of like 0.994 for viral MSA compared to 0.997 for pairwise distance calculation. And actually, when we calculated phylogenies, the phylogenies inferred using the viral MSA multiple sequence alignments were actually slightly higher topological accuracy than those estimated from MAP. So negligibly so, but still what we're showing is that these are kind of essentially equivalent in terms of accuracy for all intents and purposes. So the conclusion, viral MSA, it's a tool that enables rapid multiple sequence alignment of ultra large viral data sets. It's open source. You can find it on GitHub. And please consider using it in your viral analyses. And acknowledgements. I want to thank Heng Li. He's the developer of Minimap2, and it's really his expertise in developing Minimap2 that enables viral MSA's speed and performance. I want to thank the NSF grant that supported it, and the research was also supported using Google Cloud Platform research credits. Maybe I'll save time for any questions, or I'm happy to end it here.
Thank you so much, Niema. What I want to do first is conclude this series of presentations by thanking you and all of the other speakers for sharing your research with everyone today. And for the audience, I'm going to, it's my turn to share my screen again. So I'm going to say to the audience, each of our speakers' presentations will be made available on our website and on our YouTube channel later this week. I also want to plug that written transcripts in English and translations into Spanish will be generated by our students in the coming week as part of our ongoing accessibility and Spanish language resources initiatives. So you can find copies of all previous lightning talks on our website via the link that I'm, of course, about to share in the chat. And before we segue into the Q&A session of today's webinar, I want to briefly plug all the ways that you can stay in touch with the COVID Information Commons. So please check out our website, subscribe to our newsletter. You can also follow us on Twitter, Instagram, Slack, or LinkedIn. If you have questions or suggestions for our events, you can always email us at the address at the bottom of your screen. I'm going to again share all these links in the very heavy chat at this point. So please peruse our channels at your own pace and keep an eye out for communications from us about these upcoming events and activities. So I'm going to wrap up my admin and I'm going to open it up to Q&A from our audience. We have just a couple minutes left. I think we have a few things that are pending in the chat that Florence may be able to help moderate those Q&A sessions today. Florence, anything that we want to pose to the speakers?
Yes, we do have a few questions, so let's give it a go. I see all these smiling faces. So Ellen, 1 or maybe 1.5 questions for you, if we could. How can we use your techniques relative to new strains of COVID and other future zoonotic or related viruses? And a related question, just because of your talk, was how far back were seasonal coronaviruses recorded? So I'm trying to look backward and say, when could this have started helping us, and how might it help us looking forward?
Sure. I think for the question about seasonal coronaviruses, I might defer to Jeff, who might know more about that.
Oh, that's true. Yeah,
they've been known about for a while, like decades, but
like the 60s,
they're not novel in that regard. But with regard to detecting new strains of COVID, this method is really to detect viruses you can't detect by PCR. So the way that we make diagnostic tests is we have the viral sequence, and then we design specific probes to that viral sequence. If you don't know the sequence, you can't make a PCR test. But the good thing is that's not the way that our body diagnoses viruses. Our body diagnoses viruses by sensing features that are in common to all viruses that are different from humans. So viral RNA, the genome of viruses, is different than our RNA. And we have sensors built in that detect that. That's sort of agnostic of the sequence of the virus, pretty much. So that's why this eavesdropping on our body's diagnosis is giving us different information than the test that we design when we know what the virus is. And so it kind of helps expand out to say any virus that the body thinks is important enough to fight will pick up with our screen. And then you still need to go through the process of identifying the virus. So as far as picking up new strains of COVID, probably a lot of new variants of COVID are still going to be detected by the PCR test that we have.
Okay.
And then it's the sequencing approaches that we've heard about today that are going to really tell you if it's a new strain or just the same old thing. That being said, occasionally a virus might change in such a way it wouldn't be detectable by the typical PCR test. But our test would allow us to know there's still a virus there. Let's look into it with metagenomics or some other methods that are, we don't have to know ahead of time what the virus is, and then we can find it. So
Okay.
it's really this is a strategy for picking up viruses that we didn't know were going around. That being said, if people have historical samples in the freezer somewhere from some outbreak that was never identified, yes, this could be a way of figuring out what in historical samples are there some of those samples that are worth pursuing to see what virus is there. So I hope that answers the question.
I think it does. And so the tests I have should still work with the new strains of the coronavirus. That's what I was wondering.
Yeah, no, they should, unless it's changed dramatically. You know,
Right.
if it changes dramatically and the PCR tests don't work, then this technique would still work.
Very good. And so, Jeff, over to you, because part of the questions actually were related to you, and there's another one for you. And thank you very much for that, Ellen. It was very helpful. So I think the coronaviruses were first discovered maybe in the 1960s, from what I can tell. But maybe you can give us a little evolutionary view of that. And on the boosters that you were talking about, do you think we'll have a choice as to how often we get a booster? So like if your numbers say, oh, so 6 months is better than 12. And they say, okay, we're going to do this once a year with the flu vaccine. And I say, excuse me, can I do it every 6 months? Will I be able to do that?
All right. So first question first about the history, which is we did, in fact, study that using phylogenetic techniques. I sort of only showed one phylogeny in my talk, but there was a lot of phylogenetics underlying the work we did. So thank you, Dr. Moshiri, for working on that sort of stuff. It's very important for lots of important results we get. And so, but one of the things we did along the way, although this is unpublished, it just was part of what we did along the way, was to look at what the best estimate for when these viruses diverge from each other. And they have different divergence states. And I can't summarize it all verbally for you, but what I can tell you is just give you a general idea that they don't come out as really highly diverged compared to other viruses or other deep divergences in the evolutionary history that people think. So to give you a quantitative idea, we're talking these things may be diverged anywhere in the last 2,000 to 4,000 years is probably the error bar. Unfortunately, those kind of error bars are very large for that kind of estimate. So I know that seems like a lot. The estimate would most likely be 1,000 years ago, but it could be like in the 1800s. But that's the way the information tells us. It doesn't tell us that much, but it does tell us something, and that is that these are pretty recently diverged organisms from each other. Part of the reason why our study was possible to be done, because they're similar enough that they're not going to have highly diverged differences in how they interact with the mammalian immune system. Now,
So future Earthling Martians would be interested in your information then.
I think they would. To answer your second question about choice, this is not a question for a scientist, right? This is for
it's regulatory.
I do think that it's interesting how we sort of like constrain this kind of choice, whereas there are many other much more substantial risk choices that we are allowed to do as a society. So there's very little risk to getting an additional booster. So why do we decide that that's some risk we don't want people to be able to decide to do on their own, whereas we allow people to, on their own property, drive their car at 120 miles an hour? Yeah, I just don't really understand our relative weighing of risk versus decision-making in this society with that regard.
Yeah, I understand. I was going to be actually presenting at HIMSS last year. And I was like, okay, I want to get a booster before I go. There are going to be 20,000 people I don't know at this place, right? And so I called my doctor and said, okay, are you going to get in my way if I try to get another booster now? And she said, no. I said, okay, just checking, because they're like, well, this is the people who should go. Like right now, they say if you're over 65, I'm like, well, what if I want to get one, So I wonder how it's going to play out. So thank you for that.
Yeah, if I can extemporize a little bit more, I would also
Yeah.
just argue that unfortunately, I think one of the transitions we've had recently is from thinking about COVID-19 as a disease where we want to worry about hospitalizations only, as opposed to thinking about sickness. And quite frankly, being sick for 2 weeks is a horrible loss for me. I can't stand that, and I don't want that. And anything I can do to prevent it, I want to take action on. And unfortunately, the way our public health system has made the recent transition has really focused on stopping hospitalization and not being concerned about the enormous workforce and indeed, pain and suffering that you get from just being sick from COVID-19 or the flu or even other seasonal coronaviruses.
Yeah, and the long COVID implications. But are we going to decide there are long flu implications after we learned about this? Who knows, right? So there are a lot of implications. I totally agree. We have another question that just came hot off the press in the chat. Does age determine the immunity duration? Does it also determine the amount of frequency of which you need to take these vaccines? And this is open to anyone who wants to answer that.
So unfortunately, the only answer I can give is the statement that I probably should have finished my talk with, which is that we're looking at that right now.
Okay.
Obviously, it does have an impact. If you have a lesser immune response, you have a lower antibody level typically in response. Therefore, your durability of immunity is shorter. So if you are people who are aged have a lower immune response typically, those who are immunocompromised, those who are taking cancer treatments, et cetera, et cetera. So for those individuals, higher frequency might be warranted. We're looking into that now to get a quantitative result.
Very good. That was very helpful. So let's switch a little bit and go to Cheng Yu, CY. I'm an aerospace engineer, so I love talking about droplets and aerosols. So did a lot of work in a wind tunnel when I was a kid. And so I know that people that I know that did not leave their apartment in a very large apartment building, specifically some of my friends in India, they said that they did not leave their apartment, but they contracted COVID. And so what you were looking at, I felt like it was more like if the human who has it, goes to these other rooms, it could, of course, help, someone else get it, which is a bad thing. But did your work also relate to positive diagnosed patients in the same building that actually went through the HVAC system or other vehicles enabling transmission? Did you look at that as well?
There have been quite a few studies on that. So one study published by my colleagues was, if you are taking a ride, and if someone is sick, that's a very small indoor space, and the chance of getting the exposure is very high because it has nowhere to go unless you have a super high ventilation. You open all the windows, but that doesn't really happen in the United States, at least not in Florida. So, and there have also been studies using the simulations, computational fluid simulation, or other types of measurement to show that ventilation plays a very important role. And if you don't do it correctly, it actually can help transmit the virus from one space to the other. You're not thinking about that, but it's very likely that you don't have anyone sick in your space right over there, but because in the same building, so the ventilation, the HVAC system or other things, then the virus is very smart. They can find their way to go to your space. So, in that case, the personal protective equipment will be very helpful to reduce the risk. So yes, there have been quite a few studies on that. And
that's
unfortunately,
helpful.
yeah.
No, I'm sorry. Unfortunately, what? I didn't mean to cut you off.
Unfortunately, the virus is smarter than who we think we are. They can find their way.
That stinks. Those little suckers are so little, and they're still smarter than we are. It's so counterintuitive that if you're all by yourself and you're not going anywhere, that you need to wear your PPE, right? Like, why would people bother? And so then we're worse enemies. We don't even realize it. What you're making me think is, how they have like fire rules about, okay, if the door is hot, you don't open the door, and so I'm wondering if the air flows in a building actually, so it would be, and if there's a pandemic or people are sick in your building, don't open your door, or don't open your door. If you open your door, make sure your window is open so it goes right past you. That might be a little too complicated for normal people, but, I'm just wondering, what I would do in a situation like that, what would be better?
Yeah, on the other hand, there are other mitigations that can help us lower risk. Like during the pandemic, there were a lot of self-made air purifiers, the Corsi box, and they are quite effective in reducing the viral load in the same space. So yes, there are things that we can do to lower risk.
Well, that's very helpful. So we can keep looking at all the great research. Everyone's going to keep doing, when we started the KIC, the COVID Info Commons, there were 32 awards. Then we had like 700 and now there are 9,500, and we actually built it to scale to tens of thousands because we figured that was going to happen, but there's just so much more we still have to figure out. So thank you for sharing that.
Thank you.
So Ioannis, when you were presenting your false negatives and positives, and you said, I don't have time to explain how we fix it. I was like, oh, but I really want to know. So, so can you tell us a little bit about how you reduce the bias in the false negative and false positive cases? I was thinking there were maybe comorbidities that you didn't see at the top line in the algorithm. I didn't know what it was, but what was it and how did you do that?
Yeah, so there are a number of different approaches. So one would be to develop separate models for different parts of the population, but that unfortunately doesn't always resolve the bias issue. So one essentially can use different decision thresholds for different cohorts of the population, and you can therefore control the false positive rate and the false negative rate to the extent that you would like. Decide some way, and perhaps one reasonable way is to fix the false negative rate to some given percentage, and then set appropriate decision thresholds, so that a decision threshold is a value that you compare the score that the algorithm is giving you in terms of making a positive prediction or a negative prediction, and that threshold is something that one can adjust for the algorithm. And you could use different decision thresholds, for instance, in my example, for Black individuals and for white individuals, and achieve the same false negative rate and achieve pretty close ratios of false positive rates over false negative rates.
Are you going to be publishing these different techniques that you used or will be using?
So some of this is in our GEMIA paper that I referenced in the slides, and we have done work in other contexts. So, for instance, looking at hypertension, so those that have uncontrolled hypertension, and we have submitted some papers, and so there are more papers coming in the pipeline.
That's good because we also have a national student data core that we host through the Northeast Big Data Hub, and we have a whole data science ethics area, and we try to teach the students about the ethics of data science. And it actually starts before you even create an algorithm, like what data are you going to include and what data are you going to ignore or throw out, what's on the cutting room floor and how did you decide that, who decided it, and does everybody get an opinion? And then of course the algorithmic side and the other choices. So anything you can do to publish any of this that we could reference would really be wonderful. So I don't know if we have a link to that paper. Did we get a link, Lauren, to the paper that Ioannis is mentioning? Or maybe you could put it in the chat, Ioannis, would you mind?
Yeah, I will. Yes, definitely.
Very thoughtful. Okay. And then Niema, as you were speaking, I was wondering, can quantum computing help with your compute challenges? I know that when I was working on parallel processing and, the blue gene computer, I was at IBM for many years. We were able to make things go a lot faster and when we use new computing paradigms. And so you already found some ways to get around it because you're brilliant and fix things, which is great. But are there other mechanisms that you'd like to try? Like would quantum help, do you think?
Yeah, absolutely. So I actually, so my background is primarily in algorithms and more like on the CS theory side of things. But I've been partnering with a faculty member in my department who focuses entirely on hardware acceleration. So, so not directly answering your question yet, but I think so we have just how do you come up with better algorithms? Then, once you have the better algorithm, how do you write better CPU optimized code? Then given CPU optimized code, okay, well, how do I write? Maybe GPU optimized code, FPGA, ASIC, some other custom hardware. And then I would say maybe like the quantum computing side of things is maybe on the other end of the spectrum where the spectrum is kind of on one end is CPU, which is maybe less acceleration, but more attainable just because everyone has a CPU, whatever device you're watching this on
Right.
has a CPU that can run. GPU is a little bit more now niche, but most people have a GPU that they could use. Okay, FPGA, now you're getting into some territory where not everyone has the ability to run it. ASIC, and then, once you get into some custom hardware, and then once you get to like quantum computing, okay, well, who has a quantum computer that they could use? So I think we are looking towards going that route. I haven't looked at quantum computing specifically. Right now, we're just kind of focused on how can we come up with custom hardware to do this stuff.
Okay.
But I would imagine, I think for the virus stuff, actually, I think these problems are too small to really warrant quantum computing. I think there's a lot of benefits that we can just reap out of current hardware that we have.
Oh, good.
But I think for maybe like human genomics and stuff, I can imagine maybe if you have like global scale human genomics one day, that's somewhere where maybe quantum computing can help out. But I think in our current stage, we haven't really utilized the hardware that we currently have, and I think that's kind of my next steps.
Well, that's great. So then it'll be faster to do it. You have to wait to figure all the other new stuff out. So that's
exactly. I mean, already with FPGA, we have, I think, like 1,000x speed up compared to even what I have right now.
Yeah.
So yeah, you're looking at things that, run in like a snap of a finger, which is amazing.
I know. And I'm sorry we've kept you long. I have one last question, then we're going to scoot. If you have to scoot, you can. So, and Jeffrey, I think this one will be for you. So when I recently saw the mRNA-based Merck Moderna cancer vaccine, I was like, ooh, all the people that are like, oh, that mRNA stuff, it's too new, and I'm like, well, gosh, what if it could help you with your cancer? Would you be interested, or are you going to be allergic to the idea? So I'm wondering if you have any opinions about that vaccine, or maybe you worked on it, I don't know. And do you think that maybe the work that was done on the COVID mRNA-based vaccine actually helped them get to where they are with the cancer vaccine?
So
I know it's still for clinical trials. It's not out there yet, just so we know.
I'll answer the last question first, which is it definitely helped them to have a very successful mRNA vaccine for COVID. Raised a lot of money, but also just made everyone sort of realize how successful a vaccine could be. And also brought home the point, which had been sort of a selling point for many years for mRNA vaccines, but to see it in practice used on this very short timescale where you can like decide what you want to make a vaccine for, and 3 months later have, very large amounts of vaccine. Well, for a cancer vaccine where you may even be making it like individually for an individual, that's not how the current vaccine works. But ultimately, that is really extraordinary and has extraordinary promise, I guess, is all I would really say with regard to the cancer vaccine. The mRNA technology has tremendous potential going forward. I'm just a secondary viewer who's been studying data on it. I've never tested or thought about mRNA vaccines until it came up. If you don't mind me just going and showing one other thing super quickly, the question was asked earlier about the divergence times of the SARS-CoV-2. In the intervening time here, I was able to pull up my data sheet that I have on that. Again, this is unpublished data. It is not peer reviewed. I'm not actually sure if we're ever going to try to publish this. If anyone wants to work on that with us, please let me know. But it is, I do have the result right here, so I can just share so people have an inkling of what it would be.
Thank you. Ooh, real-time, real-world evidence. How fun.
Can you share that? How do I do that? Okay, share a screen.
That little share, just hover over the bottom. It's a little green box.
Got it. So this is just as I said, it's literally just an Excel spreadsheet my collaborators and I were throwing around. What you can see are 4 columns on the right. These are 4 different algorithms for figuring out what the divergence times are. And down here are the different divergence groups. So if you look at a pair, that's the most easy thing to see. This is gamma coronaviruses and delta coronaviruses dating back to, depending on the analysis, 3,000 to 1,500 years ago, et cetera, et cetera. So if you look at something simple like the 2 seasonal viruses, they're going back, depending on the algorithm, 2,000, 1,500, or 1,000 years ago. If you look at SARS-CoV-1 and SARS-CoV-2, 800, 400, 200 to 1,000 years ago. So even these 2 viruses that we think of as closely related because they happen to us, in short proximity, have hundreds of years of divergence between them. But that kind of divergence is not a huge evolutionary divergence for an evolutionary biologist.
Very interesting. Or like with climate change, will that accelerate with more zoonotic viruses? Or I don't know. When I look at numbers like this, I think,
I think, Dr. Moshiri, you have a question?
Yeah, just real quick. So I would caution, were these just kind of, you took full genomes and then threw them like through tree time and IQ tree in real time? Because I think recombination really shoots up those TMRCA estimates.
We had estimates of recombination, at least in the SARS-CoV-1 and SARS-CoV-2, and we threw out recombinant regions.
Okay. Oh, perfect. Okay, that's good to hear.
you're also compatible. Boy, I'm glad we got you together. I feel like we should have like another, Zoom party. Well, thank you. Yeah, wouldn't that be fun? Get the gang together again. Thank you so much for sharing that, Jeffrey. Very interesting.
It's helpful to people to conceptualize this.
This is all very helpful. This has been amazing. Thank you all so much for sharing your research, your brilliance, your team's work. This was really great. And Lauren, thank you so much for organizing this as always.
Absolutely. This has been a really great session, and I'm so pleased that we were able to find the time that worked for all of you.
Yeah, and if we can do anything else, there are some other collaborations that you would like to get involved in, reach out to each other, reach out to us. We're a collaboration hub and a catalyst for innovation. That's kind of what we do for a living. And we love working with all of you, and we really appreciate you being part of the KIC community. So anything else, let us know. And if not, be well and stay safe, and we'll look forward to connecting again.
Thank you, everyone.
Thank you. Bye-bye.